Thanks very much. Can you hear me? Okay, so I realize that it's an interdisciplinary crowd, and so if you find something unclear, please feel free to interrupt, ask me questions. So since this is a physics colloquium, I should start. I should get the microphone working. I should start with some solid facts that I'm sure all of you know, but just to make sure we're all on the same page, right? So we're going to think of a human brain as this giant network of 10 to the 11 neurons, each making about 10,000 synapses with other neurons, right? And the way this system works is actually rather simple. At least the components are rather simple. So what each neuron does, it collects electrical currents that flow from the synapses, and those currents are elicited by the inputs from the upstream neurons, and the currents flow into the body of the neuron, get integrated, and when they reach a threshold, a spike is generated that travels down outside of this neuron, and it's transmitted across a synapse to another neuron. And that other neuron repeats the whole process all over again. Okay? Of course, there aren't just two neurons, 10 to the 11. There aren't just one synapse. There are 10 to the 4, 10 to the 5 for each neuron. So the whole thing is rather complex. And that's what originally attracted me to neuroscience. And I think that now is actually a great time to be in neuroscience, and you sort of feel like a kid in a candy store because of the technological developments of the last few decades, which completely revolutionized the way that experiments can be done now. So here are a couple of examples. Forgot to say that there are two dynamical processes that we will worry about. One is fast dynamics of electrical activity in neurons. That's the pulses that propagate and the integration of the currents. And then there is synaptic weights, which is how much is the spike scaled for transmission into the downstream neuron. And that synaptic weight can also change with time, but a much slower time scale. That's the slow dynamics. Okay? So here is a picture of the fast dynamics, right? So what neuroscientists can routinely do is observe the brain in action. Specifically, they can genetically engineer a mouse that has calcium indicators in each neuron. These are molecules whose fluorescence changes with the concentration of calcium. And the concentration of calcium reports on the neuronal activity. When neuron is active, calcium concentration goes up. So this way you can optically monitor the activity of now thousands of neurons in a live behaving animal. So this mouse is sitting here watching a TV, and its head is under microscope. Usually it's also running on a treadmill, okay? And so it's in virtual reality. And so what neuroscientists can do, they can watch its brain, okay? There is a scale here, but basically each of those blobs is an individual neuron. And when it lights up, it means it's active, okay? And so this movie is already probably 6, 7 years old. And so now they scaled up to thousands of neurons, and they can even image areas of the size of the whole mouse brain. Okay? Of course, you can't just publish a video, at least not anymore. And so what neuroscientists want to have is a spike train that is the activity trace for each neuron. And this is a shameless plug for software package called Cayman that was developed in my group in Flatiron that transforms these videos into activity traces automatically. Okay, so this used to be a laborious process which took longer to extract those spike trains manually than to actually acquire data. And now you can do this automatically. Okay, now that we eliminated this bottleneck, we're facing, of course, another problem is now we have to understand what those spikes mean. Okay, so for the purpose of this talk, we actually will not worry about the fact that neurons communicate with spikes. We will characterize their activity with a continuous real variable, say firing rate. And because we will look at multiple neurons, there will be a vector of activity yt at a time t, components of which are activities of individual neurons. So that's a variable that we will operate with. Okay? But this is not the only kind of data that neuroscientists now have in their possession. So this reflects the function of the circuitry. But of course, that function is implemented or mediated by the network of synapses that connect those neurons. And up until recently, there was no way to know what that network is. So a lot of neuroscientists pretended that it was not important. Okay, it was kind of a black box. But we didn't think so. And so we developed a technology to actually map out this network, which is now called the connectome. And it's done in the following way. Of course, you know, the animal is not alive anymore. Its brain has been fixed and embedded and turned in basically into a block of plastic. But the synaptic structure is preserved. So when it's sliced, like prosciutto, except that each slice is about 30 nanometers thick, and then each slice is imaged under electron microscope because you need the level of details on a nanometer scale, which is the size of the thinnest processes and synapses. You get these grayscale images which show the membranes of cells. And then this coloring in operation is actually fake coloring that is done on a computer to reconstruct the three-dimensional shapes of individual neurons. Then if you identify synapses, you can trace them to the cell bodies and establish which neurons communicate with which other neurons via synapses and reconstruct the connectome or the wiring diagram of that particular part of the brain. Okay, so this is, of course, not the only two technological advances that happened to the neuroscience, but in my view, these are probably the most important at the moment. Okay? So with all these advances in technology, it became painfully clear that the problem is not on the experimental side. The problem is on the theoretical side. We really don't know what this activity of neurons means in terms of computations in the brain and how that circuit produces this activity and how the activity through that slow dynamics, through the modification of synapses based on learning rules, changes the synaptic weights, which actually we can also reconstruct through connectomics. So we know not just which neurons are connected, but how strongly. So the weights of connections are also available to us from this electron microscopy reconstruction. Okay? So what can we do with understanding how this whole thing works? Well, first let me tell you what people have done before. I can't, of course, describe every approach, but I can summarize a lot of them in this very schematic kind of picture. And I will call this the reconstruction approach, or a linear decoding view. And this is an approach to understand and interpret the activity of neurons in the brain. So here are the neurons in the visual cortex. And the idea is that they are active in response to the visual stimulus shown to the brain. And it's known since the work of Hubel and Wiesel that different neurons in this primary visual cortex of the mammalian brain are edge detectors that respond preferentially to edges of different orientation. Okay? So like in that movie that I showed you initially, the neurons will light up. When will they light up? Well, when an edge of a certain orientation crosses their receptive field. Okay? So how do you think about the activity of those neurons? What the reconstruction approach postulates is that the brain basically reconstructs the sensory stimulus in the activity of those neurons. Okay? So if you represent the stimulus as a vector x, each component of which is a pixel intensity from a given patch, then you can represent it as a weighted sum of a neuronal activity where y is that vector, remember, each component of which is an activity of an individual neuron, and w is a feature vector of that neuron. And the feature vectors of individual neurons are this so-called Gabor patches or edge detectors, as I just mentioned, as was discovered by Hubel and Wiesel. Okay? So this is like basis decomposition, right? Like a Fourier transform, except that those features, those components, don't have to be orthogonal. And in fact, they are not. And the basis is over-complete. It's more like a dictionary, as they call it. Okay? So the reconstruction approach postulates that the sensory stimulus is represented as those features, in terms of those features, with coefficients encoded in the neuronal activity. Of course, it doesn't mean that this is like a TV monitor and there is a homunculus watching it, inside your brain, right? It's just a theoretical concept that allows us to make progress with how the brain processes information without losing a lot of it, okay? So the reconstruction approach can be taken to a much deeper level where you can actually use this postulate to derive a neuronal circuit and an algorithm that is implemented by those neurons. And I will describe it a little bit more in detail in the context of our work, so I'm not going to say much about it. But basically, you just write down an objective function, which is x minus the sum squared, and you minimize it, okay? And then, as a result of this minimization, you can derive a network of this kind of form where w's, which here are neuronal feature vectors, will be encoded in the weights of synapses projecting from the input neurons, encoding pixel intensity, to those neurons in V1, which encode those features. Okay? The way this is done is basically by taking gradient descent on this mismatch squared objective function. And the gradient with respect to the activity of neurons will give you this network and this weighted summation in neurons, which we know neurons do. The derivative with respect to the W to the features will give you this learning rule, which will tell you how synapses should be adjusted based on the activity of the upstream neurons X and downstream neurons Y. There were many, many papers using this kind of approach, and perhaps the most famous and most impactful one was by Olshausen and Field, where they showed that if they minimize the objective function, which is just reconstruction error squared, plus an absolute value, sum of absolute value activities, which is what computer scientists call L1 norm regularizer, then you can learn W's. The network learns W's automatically in so-called unsupervised setting, where you don't tell the network what to do. All you do is give it image after image after image. So they showed a collection of natural images to the network with randomly initialized synaptic weights, and the network, after 100,000 to 1 million images, in fact learned this kind of features that were encoded in those synapses. And those features are, of course, exactly the same ones that have been measured experimentally in the visual cortices of mammals. So that's why this work made a major impact. However, this approach ran into some major problems, and I'll mention a couple of them. The first problem is the issue of invariance of activity patterns, right? So the reason we can all communicate with each other and we understand the same thing, we assign the same meaning to the same word, must be because there is some kind of invariance between our brains, right? But there is no sign of invariance in this framework, because if you look in the mammalian brain, then the same kind of stimulus will elicit a completely different activity patterns in the brains of different individuals. But if the activity patterns are so different, how is it possible that there is an invariance? How is it possible that when I say the word red, you will understand, you will interpret it with the same color, and you will create the same mental image of red in your brain, if it's not guaranteed that the pattern is completely different, the pattern is the same? And the second problem with the reconstruction approach is a more technical one, but it is very, very important actually, and it is what's called the non-local synaptic learning rules. So what I mean by that is reflected in this kind of update for the synaptic weight, and I kept the subscripts here to make this completely explicit. You see that the synaptic weight between neurons J and I is updated based on the activity of neuron J, activity of neuron I, which is fine because that synapse connects those two neurons, so it has access to those neurons' activities. But it also invokes this sum with as arbitrary weights of activities of all other neurons. And that's not possible biologically, because this little synapse that sits in the midst of the brain has no way to access this information. Maybe it can sense some average neuronal activity, but there is no way it can know the activity of individual neurons in the system other than the two neurons that the synapse connects. And this is a major flaw, not just with this kind of algorithm, but most of the algorithms that are used in machine learning. For example, the famous backpropagation algorithm that is used in deep learning for convolutional and other deep networks you read about in New York Times, uses a non-local learning rule, and therefore is unlikely to be a candidate for a model of biological computation. Of course, it is also done in a supervised setting, and to train requires huge data sets with millions of labeled examples, which in biology are almost never present. So that's why we decided to try to build an alternative machine learning framework, which was what we call biologically plausible, that has local synaptic learning rules and that starts out with unsupervised setting, where the task is not specified, but the network is just shown example after example after example. Are there any questions so far? Please. That too. That too. So in fact, it depends how deep into the brain you go. But if you go into the cortex, you already get slightly different activity for very similar presentations. And it's not completely clear what the cause for this is, but basically the brain isn't just sitting there and just like waiting. It's not a physical system that you can prepare in a fixed state, right? It's also thinking, doing something. And so there is a lot of variance there too.
Are these dendritic bidirectional? Because in all these things, you have arrows going one direction. Like in the previous slide,
Yes.
you had 1 and 3 going to 2 and 4.
Yes.
If these things activate, doesn't it go back?
So synaptic connections, chemical synaptic connections are unidirectional. There could be a reciprocal connection. There are a lot of reciprocal connections in the brain, but there are also a lot of connections that are not reciprocated. Okay? So there are a lot of actually architectural differences also with the deep learning networks on that level as well. Any other questions? So that's right. So if you were able to come up with a different architecture that performs the same algorithm, that implements the same algorithm, but with local learning rules, you would be fine. And in fact, there is a 2-layer architecture for the Olshausen field network that does that. But when we started comparing that network to experiment, it really didn't fit. And it's more like this single-layer network that seems to fit what happens in reality. Okay? But that's exactly the challenge. I'm not necessarily, there is nothing really, I mean, there is this issue of invariance that kind of, cast a shadow on this reconstruction view. But, right, if you were able to come up with an architecture that has local learning rules and does something useful, you would already have a viable candidate for biological algorithm. But it's not that easy. A lot of people have tried to do that by doing it heuristically. They would come up with a network. They would invent very clever rules and see what the network does. Okay? And the network could do interesting things. But we are not satisfied with that approach because whatever the network is, it has to work for all kinds of data. Right? And it's very difficult to prove that a network with heuristically designed learning rules would produce a certain specific kind of output, certain kind of computation. Right? So what can be done instead? Well, instead you can derive a network from an objective function, the solution of which is known for all kinds of inputs. Then we can hope that that network would perform for all inputs. That's the approach we are taking. Okay? All right, maybe it will become more clear as I go on. Are there any other questions? Okay. So here's our strategy. As I already hinted, maybe that reflects our background as physicists, but we like to start by formulating a computational problem like dimensionality reduction or clustering or manifold learning as an optimization problem. Then we solve this problem with an online algorithm. Okay, what's an online algorithm? This has to do with the fact that the neurons do not work like our current computers with CPU architecture and a huge amount of RAM, you cannot load all the data in the neuron and then let it crunch the numbers, right? The data is continuously streamed to the neurons by our sensory organs. And they cannot, and the brain cannot wait until the whole data set is streamed, because by that time we would be dead. So it has to output the results of the computations online, in real time, on the fly. That's why the algorithm has to be online. Moreover, a neuron doesn't have at its disposal a large, random, accessible memory. Instead, it has to compute with very limited memory capacity. How limited, we don't know for sure. We assign a real variable to each synaptic input. But certainly, neurons cannot store any significant fraction of the data set they have seen or the output they have previously produced. So they have to compute some very compressed representation of the data. Finally, when we have an online algorithm that optimizes the objective function, we derive a neural network that implements that algorithm in the two kinds of dynamic that we've seen before. One is the fast dynamic of neural activity, and slow dynamic using synaptic learning rules that have to be local. So that's our strategy. That's how we define the game. We, of course, are not the first ones who did that. In fact, Olshausen and Field and people who used the reconstruction approach did exactly that. But they ran into this problem with the non-locality of synaptic rules. So to keep this approach, we had to come up with a different objective function, hoping that it will give us local synaptic learning rules. And to justify, or to motivate rather, our guess for the objective function, I want to show you a little bit more of experimental data. So here is data which was recorded by fMRI in human visual cortex. In fact, it's higher level of visual processing called IT. And what they did, they showed to subjects in a scanner a large number of visual stimuli, like faces, body parts, household objects, at least if you live in the United States. And they recorded that vector of activity. Remember the vector of activity where each component was an individual neuron? Well, in fMRI, each component is not an activity of an individual neuron. It's an activity of a voxel that has 10 to the 5 neurons. Nevertheless, they recorded such vectors, okay? And then they computed an inner product between all possible pairs of vectors elicited by such objects, okay? And that's what's shown in this similarity matrix, as we call it, okay? So it's like a correlation, right? So, of course, the highest inner product is when you multiply the vector by itself. It's just each norm. It's on the diagonal. But then there is more structure here. You see that there are these blocks on the diagonal and off the diagonal. And they happen to correspond to cognitive categories, like animate and inanimate objects. Okay? So that tells us that brains actually care about similarity in this kind of cognitive sense, that similar kind of objects are represented by similar activity patterns in part of the brain called IT. Now, this kind of experiment was done with fMRI, so it's kind of coarse-grained. What about individual neurons? Well, in fact, a very similar kind of calculation has been done on the data recorded with individual electrodes, so recorded from individual neurons, but in monkey area AT. And the similarity of the two similarity matrices is stunning, right? So just like the representation in the human IT cares about the cognitive similarity of different patterns, so does the a representation in the monkey IT. Okay? This is incredible, right? Because we can immediately take care of one of the challenges to the reconstruction approach, the invariance challenge, right? So this could explain why we can all communicate with each other and why there are invariant representations in our brain. So it's okay that when I say a word red, it elicits different activity patterns in each one of your brains. What is crucial is that that activity pattern is correlated the same way with the activity patterns of other elicited by other similar words, such as pink and orange and blood and so on. Okay? So it's not the vector itself that is important, but it's the relationships between vectors that may be crucial for brain representations. Well, of course, this is interspecies invariance, so it's like between monkeys and humans. Maybe it's too much invariance, I don't know. But anyway, we actually didn't know about this data when we were working on our approach, but it's a very nice way to introduce what we did. So we said then, okay, so maybe what the network cares about is not reconstructing the input with the output, but preserving the similarity of the input in the output. Okay, so to be more precise mathematically, these are different vectors corresponding to different stimuli in the input space, the output space, which can have different number of dimensions. We characterize similarity of a pair of inputs as an inner product. And then what I'm suggesting is that the matrix of all pairwise inner products, the similarity matrix, or a Gramian if you're a computer scientist, okay, is reproduced in the output, okay? The similarity of the output patterns matches the similarity of the input patterns, okay? Now, it's not a covariance matrix in a usual way, right? Because the covariance matrix has the number of dimensions n by n, right, or k by k. Here, the size of the matrix is t by t, where capital T is the number of examples that you've shown. And it's the same between input and output. That's why I can match those matrices. Okay? So, of course, because we want to use the optimization approach, we have to write down an objective function. And so here you go. Instead of the reconstruction, minimizing reconstruction error, we minimize the squared mismatch between the two similarity matrices. Okay? So here we sum over all the examples, and these are all the elements in these two matrices, and they minimize over the output. Okay? To avoid the trivial solution where y is equal to x, we require that the number of dimensions in the output is less than the number of dimensions in the input. In fact, this is a well-known objective function used in multidimensional scaling, and it's known that the solution is principal component analysis, or just projection onto the principal subspace of the input covariance matrix. If it's not obvious, you can think of performing a singular value decomposition on x's and y's, then convincing yourself that the singular vectors should be the same to minimize the objective, and then should match, and then the singular values should match as well. Okay? So this is the objective function that we came up with for our similarity approach. But you might be wondering, how could this possibly work? Because I made a big deal about the fact that the algorithm that the neurons implement has to be done online for the streaming data. Okay? Here, you have a summation, two summations over time. So all the pairwise interactions between different stimuli have to be involved in computing this objective. So how can neurons possibly do it if at any given time they only have access to the current stimulus. How can this objective function be optimized online? And this was one of our major contributions, that we came up with an algorithm that would do that. And this slide is a little bit formula heavy, but it's a physics colloquium, so I can do that, right? Basically, okay, so here's this objective. This is our optimization problem. And we're going to optimize it online as data is fed one sample at a time. To do this, we're going to expand the square, okay? And the first term that actually includes the optimization variable y is the cross term, quadratic in y and quadratic in x. And we can massage this term by just rearranging the order of these vectors, and so that the summation over one of the time indices is all included here. And then we can rewrite it using this trick in terms of this dummy variable wyx. And this is akin to a transformation known in physics as a Hubbard-Strutanovich transformation. Okay? But if you don't know what it is, don't worry. All you have to do to convince yourself that this is true is to take a derivative with respect to w, set it to 0. Then you will find that w is exactly this. You plug it back in, you get back this expression. Okay, so it's just a mathematical or algebraic trick. But what allows us to do is to get rid of the second summation. And now this kind of optimization can be carried out online. The same kind of trick can be applied to the second term involved in y, which is quadratic in y. And together, it gives a very simple iteration scheme where at each time step, you perform this iteration step, which quickly converges to find the output yt in terms of the input vector xt and this double variable wyx. Where does that variable come from? Well, it gets updated on each step as well based on these learning rules that you can simply derive as a recursive relations for this sample covariance sum. So these w's are just sample covariances of input and output and sample covariance of outputs. And you can update it using these terms. What's the big deal about this? The big deal about this is that this iteration that computes the output yt can be implemented in this neural network where wyx are matrices whose elements are encoded in the synaptic weights of the feed-forward projections. wyy is a matrix whose elements are encoded as synaptic weights of those lateral connections with the minus. But what's most important is that these learning rules now don't have this annoying sum and are in fact completely local, meaning, again, that the update of a synapse between neuron I and J depends on the activity of only those two neurons, I and J. So no communication over long distances is required, and it can be done with well-known biological mechanisms that are well established in so-called Hebbian for feed-forward and anti-Hebbian for lateral connection learning rules. Any questions?
This lack of long-range correlation needs to be sort of artificial, right? It still needs to persist in your real signals.
That's right. That's right. There is no nothing that rules out correlations between this distant points, right? They can be still embedded in those W's. Locality in this sense means that just the rule on the synaptic update depends on the activity of only the two neurons that the synapse connects and no other neurons. Okay, so that's what locality means in this sense. But it can capture global correlations, as long as there are wires going between those neurons. Okay? All right. Good. So we achieved our goal and was satisfying theoretically, but does it have anything to do with reality? And although we don't have a definitive test, I'm going to show you some experimental data that suggests that we are at least on the right track. So one major prediction we have here is that if this objective function does principal component analysis, then this synaptic vector for the weights converging onto individual neuron should be the top loading of the top principal component, or the first top eigenvector of the input covariance matrix. Okay? And we can now test that because we have two kinds of data that I described in the beginning of the talk. One is the connectome that was obtained by, not by us, by another group in a fruit fly larva. It's a very small animal whose olfactory system has only 21 receptor neurons. Each of them is specialized for detecting different odors. But there is overlap. So when an odorant is presented in sufficiently high concentration, it excites more than one olfactory receptor neuron. Okay. Here it is. So our collaborators, the Samuel Lab at Harvard, has recorded the activity of all of those 21 neurons using calcium imaging, like I showed in the beginning, in response to a large set of different chemicals. And this is the activity matrix that they have measured. As I was starting to say, the connectivity from those olfactory receptor neurons to the interneurons in the next layer has also been completely reconstructed. And we know not just which neurons are connected to which, but also the strengths or the weights of individual connections encoded in this vector W. Then our prediction would be that this vector W, 21 component vector W, should be equal to the top eigenvector of the covariance of this data matrix. Is it? I have to admit that the agreement is not exact, but there is enough of a correlation between the two. And when doing statistics, and we can show that there is a statistical significance to that finding. So, in fact, the synaptic weights projecting from those olfactory receptor neurons onto this interneuron do correspond to the top principal component in this order data set. Now, as I said, in no way this proves that our theory is correct. It just says that maybe we are moving in the correct direction. There are many, many caveats here. For example, how is it possible that the data set we've chosen, that our collaborators have chosen, is the same one for which those weights have been trained? Okay? Doesn't have to be the case, right? But that actually kind of shows that we are incredibly lucky that we got some agreement despite knowing what the true natural stimulus is for the olfactory system. Okay? Another question is, well, this isn't the only neuron in this layer. There are other neurons that receive synaptic connections from those 21 receptor neurons. What about them? Well, we do find some correspondence with the lower principal component. But the significance there at the moment is not high enough to rule out that it's a statistical coincidence. So we don't know that yet. And finally, the other final objection I want to mention is that, well, the top principal component is not enough to differentiate PCA from any other method. Like if you did clustering and just found the center of mass of the data, for the top component, you would get very similar results. So I cannot claim that this is exactly PCA. But something of that sort is actually taking place in the system. Any questions? For a completely new smell? No, because we don't know the affinities to those olfactory receptor neurons. We can try to predict for mixtures. And in fact, our collaborators are doing that. So based on the pure presentations, we can predict mixture, but not for the new ones.
If you did a similarity matrix with the types of smells and go through, you would be able to see what kind of responses are coming from that neuron by knowing what similar cases.
Yes, yes, exactly. Exactly. So that's what you will give, that you will get here. And in fact, you kind of can make out two clusters here. And they correspond to what they call aromatic and aliphatic compounds. And the interesting thing here is that we've been searching for this for a very long time, trying to find this kind of test of the PCA idea. But we couldn't. And the reason is we were searching in the feed-forward pathway. And here it's a feedback system. So whatever is, when the principal component is extracted, it's projected back with a minus to the neuron. So it's subtracted from the system. So what's happening here is that the network is taking out these correlations that apparently it doesn't care about. That's what I think is going on. So sort of whitening of the signal. Okay, but of course, this is still work in progress. This is, okay, so I think time is a few more minutes. Okay, so I got to the midpoint of my talk, but I can just, I think this is an important point to communicate. What I want to do next is just to say where we went from here. And we basically expanded on this idea. And it turned out that one of the major features that we are kind of ignoring here is that these neurons that we derived here are linear. They just sum the inputs with weights. But actual neurons, they rectify the inputs. To reflect this in the objective function, what we did, we just required that the output has to be non-negative. That's it. Amazingly, this very simple non-negativity constraint turned this highly, easily solvable, tractable problem that gave you PCA into an exponentially hard symmetric non-negative matrix factorization problem, which may not even have a unique, may have local minima, not just a single global minima, okay? So it's very hard to deal with. But from our perspective, it's as easy to derive a network there as it is for the old one. The only adjustment we have to do is this: in the iteration step, we have to put a rectifier in. And that's what brings our linear networks in alignment with biology by making them rectifying linear units. Just to, but we can derive this from the non-negativity constraint, okay? Of course, because we don't know what the solution for this objective function is, it would be difficult for us to know what the network would compute. But actually, over the last few years, we've done a lot of work proving that in certain special cases, we can find out what the solution of the optimization problem is. And it turns out that if data that comes in has easily identifiable clusters, like here, then this network actually clusters the output. What that means is that each of the 3 output neurons responds only to the inputs from one of the 3 clouds. Of course, this input got captured by 2 neurons, but that's actually not a bug, but a feature, because it happens to lie in between, so the network did a soft assignment. In fact, this network does soft clustering. Now, in the context of general problems that are tackled by deep learning networks, of course, what's important is things like categorization, like identifying different faces, regardless of the orientation of that face. And that problem can be cast as a manifold learning problem. And this has to do with the fact that in the high-dimensional space of pixel intensities, to be able to do something useful like learn or differentiate or categorize, the data has to live on this lower dimensional manifolds that are highly entangled. And then one idea about how the brain does it is that it pulls those manifolds apart while preserving their local neighborhood structure. And what we did is we showed that our network can do the same. Although in much simpler examples like those spirals, it can actually tease them apart where neurons learn local receptive fields that overlap with each other and learn the manifolds in the correlations of those neuronal activities. And I can't resist but point out the similarity of these kinds of receptive fields that were learned in a supervised fashion with those recorded in rodent hippocampus, which are called place cells, which earned a Nobel Prize a few years ago, where individual neurons is active only in a particular part of space. And that's exactly what happens in our network. Of course, our network is not the only or the first one that could produce those localized receptive fields. The advantage of our approach is that our networks are derived from the objective function. So we can actually prove that that's going to be the solution for a particular kind of data sets. And that is what we are after. And now, actually, we were able to do this by using methods of group theory for continuous symmetric manifolds and show that, in fact, this is the case. You can read about this in the latest NeurIPS paper. So I should probably skip to the conclusions. Yeah, and actually, this is an illustration of the pulling apart entangled manifolds in a deep network, which actually does happen with our networks if you set them up right. But of course, we are not up to the level of, solving this problem of pattern recognition for real-life visual stimuli. That's still work in progress. So to summarize, what started out as a drive to satisfy this annoying biological constraint of local synaptic learning rules, an approach we call similarity matching, turned out to be extremely powerful for constructing biologically plausible networks that now form a big family, and those networks can do dimensionality reduction, clustering, and manifold learning, which basically changed our way of thinking about biological constraints. Rather than thinking of biological constraints as a nuisance and sort of, fighting with one arm tied behind your back, these biological constraints actually are helpful, we think, because they guide the development of our theory. And by reducing the volume of the theory space that we have to explore, they actually help us to come up with new algorithms that we hope will one day be competitive with the machine learning algorithms you read about New York Times. I actually should say in that regard that our dimensionality reduction online PCA is competitive with the state of the art. Okay, and that kind of puts in the perspective of what the group does. It's a sort of a closed loop between machine learning algorithms and experimental neuroscience data. And these are past and present members of the group that were involved in this work. And I want to thank them for their contributions and you for your attention.